So I will be teaching virology and immunology. When we first started the Wits RHI course 2 years ago in the Eastern Cape, we had to decide who's going to teach what, and Dave Stead will do some of the cases, and we had Dr. Kip doing some of the pediatrics, and the thing that was left over in the end was immunology and virology, and that's why I got it. I am not an expert on immunology and virology. I've learned a lot in the last 2 years because I think I've taught this presentation about 10 times by now. But it's still very much from a family physician point of view. And what I want you to know is the kind of information you will need to be able to understand HIV resistance, be able to understand what's happening in your patient when they get sick. There are definitely people in this room that know a lot more about immunology, virology than I do, and I include all the second-year interns in that. The last time I did varsity immunology and virology was about 25 years ago. So feel free if there's things that you can add, please do. If you have questions, we can ask Dr. Madeleine. So it's good to have you here. And I'm very happy for people to get inputs. If there's important, essential things which I don't know, I'm quite happy to go and look things up, and then we'll post it later on the WhatsApp group so we can have a parquet board for any questions. So this is just a quick couple introductory slides. Our first slide there is interesting. This is where it all started. 1981 in Los Angeles, when the medical world suddenly noticed that there was 5 young men who all died in different hospitals from pneumocystis carinii pneumonia. They were all homosexual, but they had no other relationship with each other whatsoever. And as you know, in our HIV negative patients, PCP, what was then called PCP, it's now called PJP, was relatively rare, and people started saying for the first time, you can't properly see it, the possibility of a cellular immune dysfunction. And it's quite interesting when you look back what it must have been like to have stood there and watched this illness arise as it did. So the person who finally managed to describe it was a woman. I can never pronounce her. My French is not great. Françoise Barzinossi. She's still alive, a French virologist. And she found HIV type 1. We'll talk about the different HIV types. And was able to finally identify the virus that was causing all this trouble. So one of the things they do like, usually in the MCQs and the HIV diplomas, you have to understand the classification of HIV. There are some things I'm assuming you know quite well. I'm going to go over those really, really quickly. You will also notice I skip a lot of slides because I'm expecting you to read. And also we want to focus on the things I want you to take home, not on all the academics. So with the classification, there are 2 types of HIV that's been described. They're like a little bit like the wolf family and the dog, the wolf and the dog. They both belong to the dog family, but they're actually quite different. We're not going to spend a lot of time on HIV type 2, but HIV type 2, there's always questions that come up. So there's a few things I'll highlight. HIV type 2 is different kettle of fish than HIV type 1. It's not as serious. It does not progress to AIDS. As a matter of fact, between 85 to 95% of patients never progress to AIDS. It's got a very slow effect on the immune system. It's not very transmissible. So in one of the PMTCT studies, there was no events of it being passed over to the baby. And because it's not easy to pass on, because you don't get very sick from it, and because you don't pass it on to your babies, it's pretty much stayed in West Africa. So the only times you're going to be worried about HIV type 2 here is if you've got somebody who's actually traveled from that area. It's not yet an illness that's progressed down to South Africa. What's interesting is that our drugs, some of our drugs work for HIV too, but nevirapine and efavirenz does not. So those patients will be naturally resistant to our first line. And when we do the new fancy type 4 HIV testing, the VCTs, they do test for 1 and 2. But the viral load, interestingly, is different. The viral load is specific to HIV type 1. So if you did a viral load on this patient, the viral load will be LDL. And we recently had a case of somebody who's HIV positive, but the viral load is LDL. And then it's just something, who's she married to? She's from somewhere else. We're not going to spend too much time on that. What we do need to know is the classification of HIV. It's got a genus, the lentivirus, which means slow poison, and it belongs to Retroviridae. I'm quite sure you all know about HIV's amazing ability to do this thing called reverse transcription. So just to remind you what reverse transcription means, the majority of organisms, when they replicate or when they certainly pass themselves on, they will pass on the information through DNA. So DNA is your recipe. It's the pattern. And the pattern is what we normally pass on, and the pattern is usually pretty correct. The RNA is like the 3D printer. Have you seen those 3D printers at the shows? They're very posh. So you put a pattern in, and it can print the particular amino acids or proteins that needs to be printed. And what's interesting about HIV is it doesn't pass on DNA, it passes on the 3D printer. And then it uses your own body cell mechanism through transcriptase to write the actual pattern. So it goes backwards from the 3D printer to the pattern. And that's a very odd thing to do. There's both good things and bad things with that. The good thing, one would say, is that it makes a lot of mistakes by doing that. Ah, good evening. You're welcome to. Jenny Nash is also one of our facilitators. Welcome. Nice to see you.
We'll see who I can recruit for my hospitals for next year.
Ah, yes. So Jenny Nash is our DCST family physician in ADM. You're going to be working with ADM. She'll be the person who will be coming around your facilities and looking after you. Very nice. Nice to see you. So the RNA goes back, it makes this pattern, makes lots of mistakes. So a lot of HIV viruses never see the light of day because the mutations don't survive. But we also know that because it makes lots of mistakes, there's also the chance that one of those mistakes can be super powerful. You've all seen X-Men? We're going to talk a lot about X-Men. So those are the mutations or the mutes. And your mutes can possibly be crippled, but your mutes can also potentially be resistant to your virus. So it gets even where the classification goes further. So we've got HIV-1, HIV-2. We're not going to think about HIV-2. HIV-1, there's various groups. The other groups are really non-significant. An outlier group, a non-M, I don't know who names these things. A non-M, non-O or mu group, that's group N, and then there's group P. You don't even have to think about those. You need to know about group M. And then under group M, there's various subtypes. And in sub-Saharan Africa, we're partly fortunate that we have predominantly one subtype. Does anybody know what that is? All of you know what that is? Which subtype do we have in South Africa? Got subtype C, okay. America, where we have a lot of our descriptions of the original, when HIV was first described and how HIV progresses, they predominantly have type B. And that's why I think what we see in South Africa, we still sometimes use old knowledge, especially with our WHO classifications and how long it takes to progress from one to the next. Now that slide's done something interesting. Hopefully that's the only slide that's done something interesting. So this is, you can see here our prevalence C as the majority. And then you will also see them talk about CRFs. These are recombinant ones, and they will say that's a combination of A and G, or a combination of B and C. And your epidemiologist will use that to help trace how the HIV virus moves around. There's just a nice slide to show us it's predominantly C here in sub-Saharan Africa, but you can see if you move north, there's much more variety of the different subtypes. And that can make it a little bit more unpredictable in terms of how people are going to respond to treatment, but specifically about prognosis. Certainly some of them are more pathogenic than others, and unfortunately C is one of the ones that progress quite quickly. Do you remember in America they always used to say it's about 10 years, you're well for 10 years on HIV and then you get sick. We don't see that in South Africa. You can see quite young people who must have contracted it within the last few years and is already sick with AIDS. So where does HIV come from? Been engineered by CIA. There's lots of stories about which you all know where HIV comes from. Oh, it's a shy group. That's all right. So we know that this is a virus that was initially a virus that infected monkeys, and just like swine flu is a virus that infected pigs and then became a human virus. So HIV used to be a virus called SIV, which still exists within a variety of monkeys. I'm not going to help you. I'm not going to trace. We'll show a little bit what the family tree looks like, but you don't have to trace on which type belongs to which monkey. It's not important. So the question is, when did that happen? When did that jump happen between the monkey population and the human population? The other question is, how did it happen? How did it go from monkeys, because it's a blood-borne disease, to human beings? Does anybody know, how did that happen? Not that we had sex with monkeys. How did that happen? How did human beings get a blood-borne disease from monkeys? You're just tired. So it's mostly through hunting. So monkeys is a major form of meat. And hunters would go, they might have cuts on their hands, there'd be a lot of blood when you are slaughtering fresh meat. And so they got infected with this SIV, which they managed to mutate to live within the human environment. And what's interesting, within monkeys, it doesn't cause AIDS. It lives fairly symbiotically in monkeys, but in human beings, the mutation has unfortunately been decimated us. So there is, I was very academic now, we can go through them all. I did not put that picture in, but I think we should replace it with Trump. Sorry. But this is just to show you that these are two different families. You've got HIV down here, and it's like the dog family, and up there is the wolf family, is HIV-1. So this is just some of our wanderings around how long has HIV-1 been around. And some scientists are quite amazing things. When they are puzzled, they do keep samples. And we actually had samples of patients who died years and years ago, where academic institutions will keep samples. And now what they've done is they've actually gone back and they've tested those samples of patients who died where it wasn't clear what actually caused the death. So, for example, there's a sample taken in 1959 from an adult male in the Democratic Republic of Congo. There's another one here from an adult female that showed already HIV in the Congo. And what's interesting, they've also got tissue samples from an American teenager who died in St. Louis in 1969. So it's probably in the last 50 years that this jump already happened. And then we had this massive, it finally grew into an epidemic, which one can have long philosophical discussions about in the early 80s. As you can see, there's a Norwegian sailor who died around 1976. It was in the sailor population that might have partly helped to spread it into a global pandemic. All very well, we're going to move on. Right. The thing you have to know is you have to know exactly what the virus looks like. You have to know HIV virus, you have to be able to draw HIV virus, you have to be able to label HIV virus. So these are one of these things we're going to draw. If I repeat myself endlessly, because I'm hoping to teach you everything you need to know about the virus. It's round. It's not just the pictures that they make it round. They are. It's actually a very round little thing, little virus, and it's very, very small. So you can fit about 10,000 HIV viruses onto the head of a pin. And that's always been the challenges with viruses. Remember, our bacteria are relatively large. We can see them under a microscope. The HIV virus is extremely small. Hi, Doc, welcome. So we'll start from the outside in. And one of the things you'll hear a lot about is the landing gear of your particular virus. So if you look at the virus from the outside, you will see it has these spikes on it. It's made up of protein, and the spike at the top is called GP120. You can remember this name, GP120. You will read it a lot also when they talk of immunology and virology and CD4 world. They talk a lot about the GP120 because that's your landing gear. It's what's going to pull in that CD4 cell. Then you've got your fusion protein, GP41. That's quite a coiled protein sitting underneath the GP120. And then you have this envelope or your lipid layer on the outside. Now, what's interesting about the lipid layer of the HIV is it's made up of human tissue. So, and you'll see how it does that. So it has the previous host's membrane. So it's made up of a human matrix envelope on the outside. And then on the inside, it's got a matrix, also protein, a jelly-like matrix called P117. And the important bit is the capsid. All of this is just there to bring the capsid. And all it needs in the capsid to replicate itself is 2 RNA strands. Remember, we said it passes on RNA, not DNA. And it's got 3 enzymes, of which you already know the names because you know the names of the drug classes. Okay? These three enzymes you've got to know inside out. It's protease, reverse transcriptase, and integrase. If it has those three enzymes and it's got its two RNA strands, it's all it needs. Because what it's going to use to replicate itself is the human body. It'll use your own factories. The core matrix, the jelly that sits in the capsid, is called P24. Some of you will remember the days when we don't really use that much anymore, but you get a P24 HIV test as well, that particularly looks at that for the antibody against that part of the virus. So that's the first, and there's a nice little electron microscope picture. You can see the actual capsid. Amazing how they do these things, isn't it? But we have to go a little bit deeper. And if you write the diploma, you read this up the night before. You're not going to use it in a lot of other contexts unless you're doing research. And that is to understand actually the expression of the genome itself. So the RNA strands, remember, are made out of a recipe. And it's an interesting recipe because it consists of 3 layers, which is actually quite an uncommon thing. Normally when you have a recipe, it's one long strip that has got all the information on it. But HIV is made like a triple split like this, and it's one of the reasons why it makes so many mistakes. But in my diploma, I was asked to talk about the structural expression of GAG, POL, and ENV for 10 points. I skipped that question, and I thought, no, I'm sure we can sacrifice something for the larger good. It's good to know what they are. They might come up in the MCQs. For me, it's not worthwhile spending hours trying to memorize all the genetics of the HIV virus. Rather sacrifice those points and focus on the clinical bit. But this is just for those of you who wonder what I should have written in my exam. Ooh, I'm not sure. Is this slide in there? That's what I added. No. But you can see GAG. It's pretty easy to remember. GAG is the 4 proteins and matrix proteins. Remember I talked about P17, P24. Lots of repetition. Polymerase is your 3 enzymes, which are reverse transcriptase, integrase, protease, and then N is for your envelope. So that's the membrane on the outside. So those are your, well, particularly for the glycoproteins, the GP120 and the GP41, because remember it's going to be using your own membrane for the actual lipid layer. These are very interesting people who study the way that HIV transmits itself, the VIVs and the REVs, and the geneticists have got a strange sense of humor when they name things. So now that we know what the virus looks like, not a complicated little virus, but there's a few things that's a problem with it. It's extremely small, very difficult to see, and it's got this lovely thing because it replicates and makes all these mistakes. We'll talk again when we talk about resistance. There's always a chance that one of those mistakes, one of those mutants, are going to be resistant to a specific ARV. So that makes it a big problem. It affects our diagnostic tests. It took us quite a while to get some good diagnostic tests for HIV. But for example, if your viral load is suppressed, your PCR might even be negative because the virus is so difficult to actually find. And of course, it's been a nightmare with the development of vaccines. And I'll put a slide in the end about on this presentation on the new vaccine that's currently out. Now you all know what the virus looks like. Now we're going to see how it replicates. This is quite fun. I've got a cool video as well to show you in the end. So great, so there's our little virus. It's got its fancy landing gear. This big thing you're seeing over here is the actual human cell. So this is the membrane of the human cell, and there's the human cell here, and the inside is the human nucleus. And you can see how small the HIV virus is in comparison with a CD4 cell. It's a very small thing. It's like termites getting into your house. And when it lands, it needs 2 things to be able to get into the cell. Firstly, the cell needs to have a CD4 receptor. CD4 receptor is a bit almost like a flagpole. It's actually a very tall receptor that sticks out quite far. But it also needs attached to it a chemokine receptor. And this is a bit like the flag. Hello, I'm a CD4. It also is where the security clearance happens. We'll talk about that when we talk about immunology. And this is like the actual door, the chemokine receptor. The chemokine receptor's got names. There's 2 types, the CCR5 or the CXCR4. And this got people very excited a few years ago because they discovered that there were some people who cannot get infected by HIV. You've all heard about slow progressors, and I'm not talking here about the slow progressors. These are people who are, even when they're exposed to HIV, they never test HIV positive. They never get HIV into their bodies. And so when they've done their various lovely academic research, they discovered that some people, a small part of the population, about 5%, has got a homogeneous defect. That means none of their cells has got CCR5. So they got very excited because they went, if we can block CCR5, if we can do something about CCR5, then maybe we can block the HIV virus from getting into the cell at all, which is the magic bullet. The problem is that there's that small percentage who doesn't have it at all, but everybody else has got a mixture. And CXCR4 is another chemokine receptor. It's also a door the HIV virus can open. And therefore, they managed to find a drug, and they managed to create a drug that can block the CCR5. Does anybody know the name of it? It's called Maraviroc. It's expensive. And the problem with Maraviroc is actually got lots of side effects. It's actually quite expensive. And it only works if you only have CCR5s. So in America, they actually have to do a genetic test to check if you have CCR5s only on all your cells, and then it works in most cases, but not even all. So it is there as a drug. We don't have it. I don't think we can have it private. But it's certainly not anywhere on the horizon for the public sector. But anyway, now it gets there. It's going to link to your CD4. And what happens is, the video will show you how this changes. It's quite clever how it changes. It opens the door. And remember I said the membrane is made of human tissue? It opens and it's actually going to melt into the cell like that. And you start seeing it will actually almost just be reintegrated back into the human genome, and the little capsid gets sent ready to go towards the nucleus of the cell. So remember we said inside the cell we have our reverse transcriptase, our integrase and our protease, and the first enzyme that has to start getting active is the reverse transcriptase. And what the reverse transcriptase does, this picture is actually not great because this already happens within the human cell. The reverse transcriptase is a 3D printer, and it's now going to make a pattern, right? But the pattern that it writes, the DNA that it writes, it uses your human factory to do that. It uses you. And because it does that, it can create a piece of DNA that can be integrated into your genetic makeup. That's like real science fiction stuff. It's quite scary. And so what happens is the RNA, the transcriptase creates an RNA, we call it a pro-DNA, and now integrase comes in, your next enzyme, and what integrase does is it now is going to incorporate this piece of HIV DNA into your larger human DNA. So now your human DNA remembers the pattern. It's going to go through the RNA printer and print all your proteins. So now happily it uses your genetic material, and it uses all of your components to build itself. Very clever. There's just a picture of integrase integrating it into the actual virus. And now what's happening is that when the RNA, remember this is now the DNA, so now the DNA is making lots of amino acids through the RNA is going to, DNA is going to translate to your 3D printer, and your 3D printer is going to start printing amino acids. So now making the chains as per the recipe from the HIV DNA, and you now get lots of long pieces that needs to be cut and measured into HIV virus. And the thing that does the cutting and the measuring is protease. So there you see very simply how the three enzymes work together. You've got your reverse transcriptase to make your DNA, your integrase to put it into the DNA, and then you get your protease that's now going to cut it and help for this virus to mature. So it creates lots of, you can see there's your gags and your ends and your, it makes all your different pieces. Together they're going to gather with the RNA. You can see they're gathering there at the cell membrane. And now again, it's going to take your own cell membrane, it's going to gather within itself, because it's now made, all it needs more RNA, it's made those. It needs the 3 enzymes, it's made those. And now all it needs to do is bud off. At this stage, it's still an immature HIV virion. Oh, and there's just a picture of the HIV budding off the surface of the CD4 cell, of the human cell. So yeah, so it's quite, you can imagine how functional that CD4 cell is. Because basically it is being used, all its energy and all its mechanisms are being used. So if you've got a patient with a CD4 count of 200, and they've got a very high viral load, all of their CD4s are looking like that. So it's not a very functional CD4 system. If you've got somebody with a CD4 of 200 and the viral load is suppressed, it's actually a completely different picture immunologically, because at least those viruses are not so beleaguered. This is just a nice slide to show the maturation, and they've even got an electron microscope picture of that, of how all of this bits of amino acids finally get arranged again in the perfect HIV cell that you need. And that's within the virus, again, protease that's busy helping that virus to mature. Am I going too quickly? Right, so I'm going to just show you a little video of